had your first gene, which was your allele, which was dominant. So now you are having recessive allele here. So you are branching it, your recessive allele from first gene with a recessive allele from your second gene, and then you have round. So one dwarf yellow round, and then one dwarf yellow wrinkled, one dwarf yellow wrinkled. And then the last one dwarf green and round. And this is homozygous recessive, one dwarf green wrinkle. So is it clear, or hello? Is it clear?
Yes, sir.
Yes, sir.
Okay, so basically it allows us to calculate and predict our outcomes very easily, and especially when it comes to breeding, it's very important that we need to know the unknown phenotypes, and that's where the test cross comes in very handy. And we can use this method to analyze the results of a cross between multiply heterozygous individuals and multiply homozygous individuals. So you have heterozygous and you have homozygous individuals here. So what basically this tells us that that ultimately the phenotypes of offspring of this cross can, if even they depend on the heterozygous parents which are transmitted from heterozygous parent, can be predicted using test cross. So this is the take home message. So any question on this so far?
No, sir. Everything is clear.
Okay. So now we are going to go to the third method, which is a probability method. And probability method is alternative to Punnett square and fork line methods. And it is based on principles of mathematics. So basically, it's like a like when you toss a coin, your outcome is always going to be one half, and Mendelian segregations ratios are nothing but same. So when you produce your heterozygous gametes, for example here, when we are crossing this heterozygous genotype with heterozygous genotype, so this is, we are intercrossing F1 into F1 and then obtaining a gamete after fertilization, and then you have these male gametes on the right side and the female gametes on the left side. So what it tells us that every gamete contributed by either male or female, it will have a probability of dominant or recessive allele one half. So if your dominant is one half, the recessive will be also one half. So it's a probability like a tossing a coin, whether it will be head or tail. So it will be always 50%. So the question comes, so can we use these frequencies to predict the outcome of our crossing two heterozygous? When we have this F1, which is heterozygous, and then we are crossing is to another heterozygous. So can we predict the frequencies based on this probability? And the answer is yes. But how we can do it? So basically, what we do, so we have the gametes which combine randomly, means they are independently assorted, and then after fertilization, they are assorted to the next generation. Sorry. Then what we do here, basically, we know that the dominant allele have a probability of 1/2. So I think there is some, which is 1/2, and then the recessive has 1/2. So what we do, basically, we take one gamete from your female side and then the another gamete from your male side, and that's how it allows us. 1 second, I think there is some error here. Basically, it allows us to obtain these frequencies using heterozygote. And once we obtained these genotypes, so we got homozygous dominant one fourth because you had your big A one half crossed with your another big A. Deepak, 1 second. I think there is some error on my part, and just give me a minute. I think there was an error with my mouse. Can you hear me?
Yes, capital A, small a, 2 by 4.
Deepak, can you hear me?
Yes, yes, yes.
Okay, sorry that there was an error with the mouse. So basically, what we can do here, once we get these genotypes and then the phenotypes, we can use these dominant hypothesis, and that dominant hypothesis can allow us to see whether we can um, predict our outcome of genotype. So this is about different methods to predict outcomes. So is there any question on this?
No, sir.
Not yet.
Okay, okay. So now we can move to the next one, which is Mendelian principles in human genetics. So as we know that the Mendelian inheritance refers to the inheritance of traits controlled by single gene with two alleles, one of which would be dominant to the other. And as we know that not many human traits are controlled by single or two genes, but there has been a good starting point.
No, I breaking.
Uh, Hello,
Yes, now it is clear.
is it? I think there is a noise from the other side.
Just a minute. Uh, so, Amol, unmute yourself. Amol, unmute yourself. Amol, Amol, I am audible. Unmute yourself.
Yeah, yeah, but I think there is a lot of noise from the other side.
No, no, now there is no noise, so you can start it. And share PPT. Yes.
Okay.
Make it a
So
make presentation mode.
is it
Yes, it.
good?
Okay, okay, good, good.
Okay. So basically, when the Mendelian principles were rediscovered in 1900, so that's where people started to think whether these Mendelian principles can be used in human genetics. And so there are several reasons that human genetics or we cannot really apply Mendelian principles as simply as in plants. And the reason is that it's very hard, or there is no possibility to make the control crosses with humans. And human heredity needs to have like a strong pedigree records, which is oftenly incomplete, because you need to have a record from generation to generation, and if you need to keep that record, it's going to have a possibility that there will not be any full record for it. And also in humans, we cannot produce many progenies like other experimental organisms, which really makes it difficult to discern the Mendelian ratios. And we cannot maintain or observe ourselves in a controlled environment. However, there have been great deal of efforts being taken by a lot of human geneticists, and especially since the human genome has already been sequenced. It has been really great development, and there have been a lot of advancements in human genetics, and application of Mendelian principles have been widely used in human genetics as well. So what we are going to do, we are going to see one trait from which is inherited by the dominant allele, and one trait inherited by recessive allele. But these are some of the examples which allow us to see what are different genes which are causing different dominant traits. And there are popular examples like dwarfism, earlobe attachment, short fingers, and one of the very well discussed is Huntington disease. So what we will do for today, we are going to see this Huntington disease as a to understand how the inheritance of dominant trait happens. And then there are other traits which are inherited by the recessive allele, like albinism, alkaptonuria, cystic fibrosis. And for this class, we are going to see the sickle cell disease and how the inheritance of sickle cell disease happens in humans. So basically, we will have one example for each, one for the dominant trait and one for the recessive trait, and how their inheritance happens in humans. So that we will see in next few slides. But before we jump into that, so we would go into the pedigree analysis first. Like, this is very important, because without knowing how to establish or how to interpret the pedigree, it will be very hard for us to do any pedigree analysis. So pedigree is nothing but a diagram which discerns the relationship among the members of the family. And conventionally, it has been used, or it has been customary, that the female parent has is circled by the circle, and then the male parent is represented by the square. And the horizontal line, I I hope I won't go to next slide with these. So the horizontal line that connects the male to the female line, that's also called a marriage line or the mating line. And then there is this sheep line where the siblings are being presented. So in this figure, I would like to explain that how the pedigree can be understood. We have three generations here: first generation, and then second generation, and then the third generation. So the first generation is for the grandparents, and the second generation is for the parents, uncles, and aunts, and then the third generation is for the siblings. In any pedigree analysis, so the branching of different levels is shown by these Roman numericals, and the number of progenies are shown by the Arabic numerical. So if we see here, your first line is for the grandparents or the first generation, second one is for the parents and uncles, and third one is for the siblings. So how we can understand this is very important, that your eldest kid will always be on the left, and then the next kid will be on the second, and then third, so on and so forth. So how we can understand the pedigree chart? Basically, the Roman numbers which are presented here are to show the generations, which we can see on the left side of the figure, that 1, 2, and 3. And so the numbers or the Arabic numbers shown here are basically showing that the individuals within a generation. So we have three generations, and in first generations we have six individuals. In second generation we have total 13 individuals, which are represented in the middle row, 1, 2, 3, all the way up to 13. And then the third row represents your third generation. So basically, this allows us to understand the pedigree chart. And one of the point I would like to mention here is that the first person in a family where the genetic study has started is known as a proband, where we can see this P shown here for the number 4 in the third generation. So this is male, so we call this propositus, and then there is, if it's a female, it's proposita. So means you don't need to know all these different things. What you need to know that the Roman numericals show the generation, Arabic numericals show the individuals within a generation, and what is a marriage line or the mating line, and ship line. So it is important for us to understand the inheritance of the trait. Is that clear?
Yes, student, it is clear.
Yes, sir. Yes,
So,
sir.
so different symbols are using to for
Yeah.
showing the pedigree.
Sorry for the cursor. Otherwise, I would have really liked to explain it in a more
No,
sophisticated way.
no, go for the left side.
Okay. So basically, this is the marriage line, and this is a ship line, and this is a proband here. So that allows us to understand how we can calculate the pedigree. So just know this. This is the generation number 1, 2, 3, and these are number Arabic numbers for indicating individuals within a generation. And usually, when we create the pedigree, you know what happens. For example, just think about if we want to see the pedigree of the 5 generations before us. So we might know how many children our grandparents had, means our parents, then uncles and aunts, and we might know even how many grandparents we had, like our grandparent and their cousin, their brother. But if we go to the great grandparent or great great grandparent, so we will not know the full details, like how many kids were there, or even if we know how many kids were there, we may not necessarily know the sequence, whether the first kid for the great great grandparent was a girl or the boy. So if the sex is not known, in that case they put this diamond sign. So basically what this diamond sign shows is that there is no sex known, what kid, whether it was boy or girl. So basically that's what these different signs show. And so we are going to go into very specific examples for the class, because we know according to Mendelian principles, each gene has one allele which is dominant allele and another allele which is recessive allele. And how that inheritance happens for these dominant allele and recessive allele. In this case, on this slide, you can see there is an inheritance of a dominance trait. So we are looking into this dominant trait here. And what we are basically doing here is we are going to see how the inheritance of this specific dominant trait happens. Because dominant trait is very easily detectable. We have seen this from the P experiment, And that same thing happens in humans also. For example, if you have a hitchhike thumb, or if you have a attached earlobe, so attached earlobe will be very easily detectable. You can find it in each generation, and usually every individual who is carrying the dominant allele that can express the trait very distinctly, and it is very easy to trace the transmission of this dominant allele through the pedigree. And every individual who is affected are anticipated that their one of the parent was affected with this trait, unless there was a mutation. But as you might recall from your previous mutation lecture, that the frequency of mutation is very low, like 1 in a million or 1 in 10 million. And conventionally, several studies which comprehensively understood the inheritance of the dominant trait, they have found out that the dominant trait has a reduced viability and fertility. And due to that feature, the dominant trait has never been a frequent in a population. So that's why we don't see a lot of people with this Huntington disease. So this is basically explains us how this inheritance happens in a overall the population, and why we see some phenomena very common and why some phenomena are not common. And we are going to see this Huntington disease, how this inheritance happens. And mostly the inheritance of the dominant trait or even recessive trait is related to the autosomal chromosome. And I'm pretty sure that you may have had the opportunity to understand what is autosome and what is sex chromosome, non-sex chromosome. So this would be very important for us to understand that autosome is not a sex chromosome. It's non-sex chromosome. And in humans, there are 23 pairs of chromosomes, and 22 chromosome pairs are autosomes, which are non-sex chromosomes, and only one chromosome pair, which is the sex chromosome, and that mainly comes from uh, the male, and that determines the sex. So without going into details, so just we need to know that the Huntington disease was inherited through the autosomal dominant trait. And just to give you a brief idea and details, what is Huntington disease, might help you to understand a little bit. So it is an progressive human brain disorder that causes an uncontrolled movements. Sometimes you have emotional problems, and you lose the cognitive ability to think. And it is very common among adults, and usually it occurs in the 30s or 40s. And typical signs vary from irritability, then depression, little bit of involuntary movements, poor coordination, and not enough ability to learn new information or strictly making decisions. So coming back to the genetics part, or how inheritance happens, the main gene which is involved in the Huntington disease is HTT gene, which is passed on from one generation to the next generation. And there are one gene is, there are repeats of trinucleotides, and this CAG is very important. And on number of how many repeats are there, that determines whether the person will have the Huntington disease or not. If there are more than 50 CAG trinucleotide repeats, there is definitely will be then possibility that Huntington disease will happen. And when that happens, that event is called as anticipation. And generally, when these repeats are less than 30, and then the specific range is 27 to 35, so that individual will be carrier of that gene, but will not express Huntington, but their children will develop it. And if you see the figure here on the left, so you have the affected male parent and then the unaffected female parent. And as we know, the male parent contributes X and Y determines the sex, and the female contributes X and X. So if we see at the bottom, there are total four kids, and in that four kids, the two on the left are affected and the two on the right are unaffected. And how that happens, basically, this XA, which is basically that contributing or causing the inheritance of Huntington disease. And same way, the girl in the second position is also affected because she got this Huntington disease through that dominant autosome. And in the last two, they didn't receive any dominant gene from their parents. So that's how the inheritance of Huntington disease, or in general, the dominant gene which causes these kind of diseases or these traits. And overall, just to summarize the inheritance of the dominant trait, this condition is caused by the autosome, and which means that one change copy or mutated copy in each cell is sufficient to cause this disorder. And so this affected person will definitely have a one parent that has this kind of disease. So is this clear on the part of inheritance of the Huntington disease or dominant trait inheritance? Any question?
No, sir.
Okay, so if not, then we can go to the next. So the inheritance of
No,
the,
no. In, in this case,
can you say that again?
no.
Hello?
Yes, so go in the previous slide. Then yes, yes, here. I think the male will not affected, no? Because
Um,
X chromosome will come from the female only in boy. And
yeah, but the affected gene is contributed by the male.
yeah, but from in this children, in this boy, the Y chromosome is coming. The
Okay.
Y chromosome is coming in this boy. Okay. So from male, Y chromosome is coming, and from female, X chromosome, and X chromosome is not affected. So this child will not affected.
No, actually, it will be affected because if you see the autosome which is contributed, that has this part of the mutation. So I think that's why it's affected, and it will be certainly affected.
Okay. Then next.
Okay, so about the recessive trait, detecting the inheritance of the recessive trait is very difficult and it's not easy because the occurrence of these inheritance is very rare, and it needs um, pedigree information of several generations. However, there are large number of recessive traits which have been observed in humans, and at least there are more than 4,000 different traits which are inherited by the recessive traits. And so one of the important difference and distinction between the dominant trait and then the recessive trait is usually when the contribution of the allele among siblings will be the one half of the allele, and then the half siblings will be one fourth, and then the first cousins will be one eighth. So when only the mating occurs or the marriages occurs between these closely relatives, then and then this recessive trait will resurface into this next generation, and we are going to see this sickle cell disease example here. So uh, on the left side, you have unaffected carrier father, and then on the right side you have the unaffected carrier mother. So, and here, if you see, this is the sickle cell disease, which is inherited in the autosomal recessive pattern. So, when these two parents are the carrier, then the probability of having the sickle cell disease will be 25%. It means this will be definitely going to happen. So, this one-fourth homozygous recessive will have the sickle cell disease. And another two will be the carriers but not having any disease in them, and then the one on the left will not be affected, neither carrier nor affected. So when we see these one on the left, two in the middle, and one on the right, it typically comes into the monohybrid ratio, like a Mendelian ratio, 1 to 1. So our when we use a dominant hypothesis, 3:1. So this is what really adheres to the Mendelian principle, that it can happen into 3:1 ratio. So this is one scenario. There is another scenario on the right side. If you see the affected father and unaffected carrier mother. So if such thing happens, then there is a chance that 50% of the children's 50% of the childrens will have the sickle cell disease, and 50% of the children will carry the trait but will not have the sickle cell disease. Is that clear?
Yes, sir.
Okay. Any question?
No, sir, not yet.
Okay.
Sir.
So then there is a little bit of extension to the pedigree analysis. I thought like this might be very interesting to think in a reality how this is really being used in practical applications. So as the advances happen, especially after the human genome was sequenced, so there is a new field coming into place, which is a genetic counseling, and most of the hospitals which have this facility. So what they do, based on the pedigree records, they know whether the individual will have this disease or not, and then they do the genetic testing. And once the genetic testing is done, and once they understand that there is a high chance of happening this, then these experts, which are the genetic counselors, they help parents in adapting and understanding how to cope up with these potential disease in the offspring in terms of medical treatments or the psychological support, or what familial implications they might expect from such a diseases. So this field is kind of gaining a lot of traction lately, and there is a lot of demand in this field. So I just thought it might help to kind of correlate how real principles from genetics are used in real life. So after that, I would like to summarize that the pedigree analysis is really useful to identify whether the dominant and recessive traits are inherited in human families and how they are inherited. And pedigree analysis really allows to identify the potential risk, that how individual will inherit the particular trait. And we have this little brainstorm activity. Just on your right there are two pedigree charts, and you would like to say which one is dominant and which one is recessive. So is there any volunteer that can tell which is the pedigree which is showing that the dominant trait is segregating, and which pedigree chart is showing the recessive allele segregation? Any volunteer?
Sir?
Yeah.
I think the first one is the recessive, and the second one is dominant.
Why do you think that?
So because in the first one, the male one is showing recessive, but in the next generation, it is present in few of them. Like in this particular pedigree, only one of them, and in the next progeny as well. So it is recessive, I think.
Okay. Right.
And
Pretty good.
and for the second one, it is dominant in male, clearly showing, and it is then passed on to the progenies to male as well as female.
Okay. Okay. Very good. Okay. Good.
Yeah.
Okay. That's what activity I had. And so, is there any question on this so far? Um,
Student, please ask question. Okay, Amol, unshare your PPT and turn on your camera because
okay.
everybody wants to see you.
Okay, so these are some of the references, and I would highly recommend you to read this book. There are, I think, 6 or 7 pages, not even 6 or 7 pages, but really good reference for what we studied. And so, hmm, can you see me now?
Yes, yes, Amol.
Okay, I would like to apologize for this cursor mishap. I think that kind of created a lot of inconvenience in switching the slides back and forth. So I sincerely apologize if it caused any inconvenience.
No, Amol, it is your hand. Uh, we have delivered a very nice lecture and it is a very complicated topic. There's a pedigree because ancestor in what is how is it transferring is very critical to understand. But you have explained very easily and simple way, and that's why our students in the brainstorming activity they have given very easily the answers of your question. So it means
Mm.
they have understand this topic, and thank you so much. And again, if you want to say anything, you can say. Okay.
Okay, so basically, I wanted to know if we should have one kind of little bit of quiz next week on the whole Mendelian inheritance for the students, not like a quiz, but just to understand the concepts. And the questions will be same from what we have discussed in the class. There will be nothing new. Just to, I would like to kind of, I like to repeat it again and again so that students understand. So it's up to you guys if it's okay.
Yes, student, are you
Otherwise,
ready? Are you ready to give the exam?
Yes,
Yes,
Yes, sir.
sir.
It's fine, sir.
Yes, sir.
Yes, very
It's
nice.
okay, sir.
okay, then I will prepare it and then I'll share it with Deepak and then he will pass on. We can have a live exam or you can have a look at it and we can discuss next week.
Sure.
Okay,
Okay.
Amol, thank you. Again, anything else you want to share?
Uh, no, I think we are good and maybe we can next week we will have cytoplasmic inheritance, I guess. So I look forward to it and so uh, let's meet next week then.
Yeah, yeah. And student, it is really happy news or thing is because cytoplasmic inheritance is a one kind of magic because you know the genes are present on the nucleus, but some genes they are present in the outside the nucleus also, and really the mode of inheritance is a magical. So not like as a normal Mendelian inheritance. So so learning this hidden fact with the guest lecture like as Amol, it is a really nice opportunity for us, and that's why we I'm requesting to you that each and every student should attend the next lecture. Really, it is a very interesting lecture. That is the cytoplasmic inheritance. And again, thanks Amol for giving your valuable time and have a nice day. Okay, thank
Me
you so
too.
much.
Okay. Bye for now.
Amol, sir.
thank you, sir.